hello and welcome everyone to our second July SIPEC seminar here at MTRI. My name is lycophyte and I'll be the moderator for tonight. I'm a research technician here at the Aquatic Research Institute. Before we begin today, I would like to acknowledge that MTRI is in and we are meeting from Keswick, one of the 7 districts of Mi'kmaq, the homeland of the Mi'kmaq people. We acknowledge treaties of peace and friendship, and we thank the Mi'kmaq people for their generosity in sharing their homeland with us. For people that might be joining us for the first time tonight, or that might have not heard of MTRI before, I'd like to give you a very short little bit of background about what we do and what we are. So MTRI is a research-based nonprofit nestled in Southwest Nova Scotia, near Acadia National Park and National Historic Site, and within the Southwest Nova Scotia Biosphere Reserve. Our mission is to promote, conserve, and sustain biodiversity in Keswick and beyond. And tonight, I would like to welcome Katie King from the Acadia University. Katie just completed her undergrad at Acadia on prototypus Quillwort, which is also the topic she'll be presenting on tonight. And she has in the past worked with the E.C. Smith Herbarium as well, the Acadia Seed Bank, and the Nova Scotia Department of Resources and Renewables, where she also helped in updating the provincial management plan for prototypus Quillwort. And in the fall, Katie will be moving on to doing her master's at Acadia as well in Alison Walker's mycology lab. Next I'll be handing it over to Katie, but I would also like to remind everyone to please keep your mics muted. And if you have any questions, you can just add them to the chat and we'll ask them in the end later. And there's also going to be a designated question period in the end where everyone can personally ask all the questions that they would like to ask. And with this, I'll hand it over to Katie and we'll start the show. Thank you very much.
Awesome, thank you very much. Trying to find a good place to stand. Thank you, yeah. Lost it again. Okay, thank you. Like Lucas said, this was a project that I had done for my honors for my undergrad, and so I just wanted to quickly thank, first of all, my honor supervisors, but also everybody who had been involved with that research or supporting me or advising me on that. And so I have quite a lengthy list of thanks to extend. And just to start, my name is Katie King, like Lucas said as well. I'm a research assistant and student at Acadia University. I just finished up my undergrad and I'm moving on to my master's in the fall. And throughout my degree so far, though, I have been working and volunteering with E.C. Smith Herbarium, the Acadia Seed Bank, and the Tissue Culture Lab, which are all located in the Casey Irving Environmental Science Center. And this is how I became involved with this species. I was working on a co-op term with the herbarium jointly with the Nova Scotia Department of Natural Resources and Renewables. And I was able to work on the management plan for the species. And that's kind of what got me first interested in prototypus Quillwort, and then from there, just learning more about it and wanting to see what we could do kind of to learn more about it is where some of these kind of projects stem from. And just to start in general, what is a quillwort? Quillworts are a genus Isoetes, which are a type of lycophyte, so they're a plant. They can be found around the globe and in either aquatic or terrestrial habitats. To look at them, like you can see in the image here, they're not a lot to look at. They're normally kind of a small, spiky rosette attached to a corm. And the bottom row is actually a different species, but all of the quillworts in this picture are really difficult to identify morphologically just by eye, which is one of the first kind of issues in working with this species. Just considering the history of lycophytes, it's in the class Lycopodiopsida, which, first of all, they're plants. So we know that they have chlorophyll, they use sunlight for nutrition, but also they had evolved at one point in their evolution to become terrestrial. So what that means is that they had to evolve stomata, they needed a cuticle, they needed some sort of coating on their spores. And so we can see that in their current day morphology, but that kind of calls back to their evolutionary history. And like I just mentioned, quillworts are aquatic. So what we know is that they evolved to live on land and then have changed and gone back to an aquatic habitat. They're vascular, so they are different from mosses and bryophytes that don't have a vascular system. So we can kind of see where they are in the evolutionary timeline because of that. But like I mentioned as well, they have spores. So they're not the same as gymnosperms or angiosperms, like the trees and regular flowers that we're used to. So kind of taking these traits, we can see where this falls on a evolutionary kind of spectrum. And that lets us know a little bit about the history of the species or this genus. And this kind of information is why we would refer to this as a living fossil. A living fossil is something that remains the same today as it did in the fossil record. So the class Lycopodiopsida first evolved around 420 million years ago. So that would have been in the Devonian, which is in kind of the blue Paleozoic era there. Whereas Isoetes, the genus of Quillworts, evolved in the Triassic, so 250 to 200 million years ago. And then by the end of the Jurassic, they had spread across the globe. So these kind of traits can let us know some things about the evolutionary success of the species, because within one era, it had kind of the evolutionary traits that it had kind of evolved to adapt to its surroundings had allowed it to spread all over the planet, and that it remains relatively unchanged since then. We can tell that those were extremely successful adaptations, that it didn't need to do much else since then. When we look at quillworts, there's about 200 to 350 extant species. So those are species that are still alive today. And that's a really large margin of error, because when you're dealing with plants, and specifically plants like lycophytes, there's a lot of confusion, which is why it's kind of a good thing to just pause and really decide how we're defining what a species is. If you think of a species as something, a group of individuals that have a genetic makeup more similar to each other than they do anything else, that they can share gametes and produce offspring that have heritable traits that they could then pass on to future generations. When you're dealing with animals, it seems like a really sound definition of a species, but when you're talking about plants, there's still a lot of gray area in here. So for example, one thing that we see a lot with quillworts is polyploidyism. So when you think of a plant, it might inherit half of its genetic code from one parent and half from the other parent. And so it would be one species with one entire genetic code. Something that plants will sometimes do is instead of just taking half from each parent, they will hold on to the entire genetic code from each parent and just double the amount of chromosomes they have. And in fact, over half of all quillwort species have some sort of polyploidyism. So I think the highest is like decaploidy. So they'll do this again and then again and again and just add to their DNA. Another thing that we get a lot with plants is hybridization. And we know that we can do this with animals, but often they can no longer produce viable offspring. Whereas with plants, with a similar kind of mechanism, they're able to then produce a hybrid species and then go on to produce viable offspring who are also and share these genetics. Or they also have the option of not bothering with sexual reproduction at all and just producing vegetatively, and then you have clones. And all in all, what I'm trying to say is that plants are really complicated, and with this species, dealing with the genetics is kind of equally as murky. So this species, prototype quillwort, Isoetes prototypus, was first defined in 1991 from a specimen found in New Brunswick in 1989. And in spite of all I just said about how very difficult it is to differentiate a species, when they first encountered prototype quillwort, they knew right away that it was something different because it is a diploid species. So what I was just saying about being able to add to their genetic makeup, this was a species that just had the two half sets of genes from each parent. So this is not a species that had many, many years of adding on to the genetic code, which is where the name prototype came from. This was clearly one of the earlier quillwort species. Since then, it had been surveyed for extensively, and it's been only found in about 20 lakes worldwide, half of which are in Nova Scotia. And so this is something that we were looking at extensively when we were trying to put together the provincial management plan, because obviously we hold 50% of the occurrences worldwide, And unfortunately, nobody's been able to actually survey these since about 2004, which at the time seemed really, really shocking that we have most of this species that's nowhere else on Earth. It's a living fossil. It could have a lot of evolutionary significance. And for whatever reason, we weren't surveying it. However, it didn't take long once we started digging into monitoring techniques and approaches to figure out why. And there's three major issues with surveying and monitoring prototype Quillwort. The first is that it's hard to locate. It's a rare species to begin with. So like I said, there's 10 sites, and while more potentially are out there, there's just a good chance that there are not a lot of them around. It's very difficult to identify. Like I had also mentioned, most Quillwort species look the same. So the most reliable way to identify a Quillwort, other than using a genetic analysis, is to look at the shape and size of the spores. And they actually have two types of spores. So these would be the megaspores, and there's also a microspore that these plants have as well. And last of all, it's hard to access. On the surface, you kind of think, yes, aquatic habitats, they're difficult to get to. We live in Canada, and we don't want to be doing field work in a lake in the cold winter kind of thing, but also even in the depth, even in summer, it's there's just so many challenges with working in an aquatic habitat. You can't lay down a grid to measure populations. You're kicking up sediment, so it's hard to see what you're working with. And these plants are usually about 1 meter to 2.5 meters under the surface of the water, so you got to dive down to check out on them. And all in all, they're just very difficult to work with. And just to discuss this a little bit more, this is a site map of the 10 known locations of prototype Quillwort in Nova Scotia. As you can see, they're very widespread. Provincially, this is ranked as a vulnerable species, which would be S3. Globally, it's ranked as imperiled. The big reason on the why where this is such a rare species, why it wouldn't have a higher classification, is because it's very likely just always been rare. If you have 20 plants and then 100 years later you still have 20 plants, it's clearly not endangered. It's just doesn't occur often. And we're curious if that is the case with the species, but not enough research has been done yet. For my honors project, I was able to do some research on this species, and I was able to incorporate surveys. So fortunately, we have updated the site surveys since 2004. So I was able to visit all 10 sites and kind of do rough population counts and just kind of check up. I was able to gather some new information about the species, just a better understanding of its biological needs and its habitat. And I was able to observe some population declines at some sites. However, we also were able to find a new site. My supervisor, Alain Beliveau, through his understanding of the species and his knowledge of the area, was able to survey site number 10, and that's a new one added as of 2022. So there's kind of positive outlook potential. As far as identifying the species, at the base of each quill, there's a small kind of little tissue pouch called the sporangium, and inside those is where the spores are held. And as you can see on the picture on the right here, sometimes there's kind of an opening so that you can see the sporangium encased at the bottom of the quill. In other, how does this say if I have my PowerPoint on? I do, okay. This area right here is called the velum. And as you can see in this image, the velum in this on the left-hand picture has grown down to completely covered down to the base of the sporangium. And in Nova Scotia, the only Quillwort species that does have a complete velum like that is prototype Quillwort. So if somebody had a specimen that they wanted to just do a quick check, they could look to see if it had this complete velum, and that could be a really good indicator that it was prototype Quillwort. However, as you can see in the picture, like the bottom of the velum is torn a bit. You can see a little bit of the sporangium, and then there's some species where the velum does extend longer. So this is not a very reliable check. And there are many Quillwort species that also have complete velums that aren't native to Nova Scotia. So just in case, this is not a very reliable check, but it's a really good kind of first step. However, this also might play a part into the distribution of the species, where in some Quillworts, the spores could potentially be more easily dispersed because they're so exposed. the complete vellum might actually hinder distribution. That's just kind of speculation, but I'm sure there's a lot of nuances with the species. The best way to identify prototype quillwort is through the spore size and ornamentation. Sorry, just a second. In this image here, I've got actually 5 different species. I don't know if you can see the letters A and D are actually the same species. They're just very different looking spore. Basically, when we're collecting any sort of quillwort and we're wanting to identify it, we need to look at the spores under a microscope. The measurement bars here are all 1 millimeter. So you can see that they are visible to the naked eye. And if you were really good with the hand lens, you'd be able to take a look at these. But basically looking at the ridges and the bumps and how large they are and the size of the spore, like I said too, where some quillworts are kind of collecting additional chromosomes, those spores actually are physically larger to hold all that additional genetic material. So the size of the spore is really important. And this is really the only surefire way to identify these species other than genetic analysis, but this is also a very difficult skill. I certainly was not able to master it and had many people help with some more difficult specimens. So, and then lastly, to access the species. So its preferred habitat is cold, clear, oligotrophic lakes. A lot of the times we found that they've been glacially formed. There's usually a sandy bottom, but underneath that, usually a calcareous bedrock. Like I said, they grow 1 to 2.5 meters deep. And along the bottom, along with sand, what we normally find is kind of an organic settlement, or a flocculent ooze, as it's sometimes called. And so many of the associated species are small spiky rosettes, such as pipewort or water lobelia or other types of quillwort. And so we're trying to look for a small spiky rosette among this while it's all covered with that kind of sediment. And here's a really, really lovely picture of prototype quillwort in situ. You can see the sandy bottom, you can see all the pipewort growing around it. And right in the center is our prototype quillwort specimen. And this is a really, really great picture. But this is much more representative of what we actually see. So if you look carefully, there are the spikes still visible, but if you're snorkeling through a lake or floating on a raft or something, trying to look for something 1 meter or 2 down, and it's covered in this ooze and this is all you have, and as soon as you get close to it, you end up picking up the sediment. It just, in doing the monitoring for the species, it became very, very clear why it was so difficult to upkeep the monitoring for the species. And so we were trying to think of, we recognize how rare and unique the species is and how very difficult it is to take track. And we were absolutely lost with figuring out how to come up with a viable monitoring solution. And fortunately, though, Kelly White from CDU, who's part of the Nova Scotia aquatics recovery team, suggested eDNA, and it's a technique she had used for monitoring endangered mussels. And basically, eDNA stands for environmental DNA. And the idea was that organisms in an environment are constantly sloughing off DNA, whether it be like skin cells or secretions, or they're dying and their cells are being kind of dispersed. And so if you take a sample of the environment, so it could be soil or air, or in our case, water, and you could apply either a species specific or a broad kind of general primer to that, it would give you information about what organisms interacted with that sample of the environment. And so the 3 kind of main applications are, you can take an environmental sample, test it against a species specific primer, and that can give you a positive kind of presence absence report. You can take an environmental sample, test it against your general primer, and that can tell you about all the biodiversity at that site. Or you can take your environmental sample and your species specific primer again, but using qPCR, which is a quantitative PCR, a different kind of amplification method, and that can give you a relative abundance of your species at that site. So all 3 of these would be extremely valuable information to have. And so this was what the core of my honors project was to try and develop some sort of approach for using eDNA to monitor for prototype Quillwort. So in doing so, I did get to, like I mentioned, incorporate surveys, but because this wasn't the core of my project, we didn't do extensive population counts, but we did get to visit all sites. We also were able to collect additional data. One of the most, in my opinion, interesting side effects, or not side effects, but side projects I was able to incorporate was the collection of an ex situ specimen. So all of this work was done under permit from Nova Scotia to be interacting with the species, but I was allowed to collect one, which we've been able to maintain in the Casey Irving Environmental Science Center. So it is still alive and doing all right. And so this can give us additional information about generation time and the length of light, or the lifespan, I should say, and lots of other biological information about the species that we just don't know. It's also been a really excellent source of DNA because as it turned out, Quillwort is extremely difficult to extract DNA from as well. So, and I just wanted to highlight this picture. This is how I spent all my summer last summer, with like snorkeling in lakes with my little floaty on my back to put my specimens in, and seaweed in my hair. And it was a really glamorous summer. So this is a really fun species if people are looking for a great fieldwork experience. So basically my project was, I collected specimens, I collected my water samples, I did my DNA extraction of Quillwort. I used DNA barcoding techniques. DNA barcoding is, instead of looking at an entire genetic sequence of a species, if there's regions that are conserved enough that they can reliably show you that this is a species, it doesn't change between individuals, but it's unique to that species. We call that a DNA barcode. It works just like the idea of a barcode scanner at a store. If we could select a region that we could use as a barcode, use that to develop a primer that we could then undergo the eDNA extraction from our environmental sample, amplify that with our primer, and we should get results. Unfortunately, because this was an honors project and we kind of ran out of time and resources, we kind of got cut off halfway through. I was able to get the DNA extracted. We were able to find a barcoding region, which turned out to be a huge hurdle as well. At the bottom of the screen, this little colorful thing, I recognize it's very small and it's okay if you can't see it, but what that is sequences from a whole bunch of Quillworts that are native to Nova Scotia. The top line across is prototype Quillwort. And one of the problems with this species, where we said it was so highly conserved, is that almost all of the DNA barcoding regions that are used for plants, for all Quillwort species in Nova Scotia, they were identical. So we couldn't use this to differentiate anything, except for this tiny little intron, or insertion, I should say, which you can see here is those white spots, that there is one little repeating pattern for prototype Quillwort, the CTTA, that none of the other Quillworts in Nova Scotia have. And it is, yeah, so basically everything had to be kind of designed so that my primer would capture that exact area, so that when we do the amplification, we were getting a region of code that could be used to definitively identify prototype Quillwort over every other species. And then we were never able to extract the DNA from the environmental sample. I think, like we said, animals would probably be secreting a lot more DNA than plants in this area. So I think probably just if we could find a way to increase the size of our sample per filtration, that would kind of deal with this problem. However, the whole process is most of the way there, and it would be great if someday we could pick this up again and continue to work with it. So in doing all of this, though, a question came up a couple times. Anytime we would talk about this species, or I would talk about my work, and somebody would invariably say, "Oh, cool, prototype Quillwort. Like, what does that do? Why is this species valuable?" And originally, I found this really frustrating because, in kind of modern terms, it doesn't fit a lot of what people like to call valuable. It's not, doesn't have any economic use, doesn't have any agricultural use. It's not a keystone species, and it's not something that we know other species are relying on. So why put all of this effort into conservation of this species that, if it were to go extinct, maybe some people might not notice? And I think this is a really dangerous perspective to take when you're talking about ecology, because one thing we need to remember is that this species evolved to fill a niche, and no other species fills that niche in its habitat or at all. And it's successfully maintained that position for millions and millions of years. And so just by evolving and being a successful evolutionary species, it has intrinsic value, and we need to recognize that. But furthermore, it's filled this niche for many, many years, and suddenly we're seeing declines in the last maybe 100 years, hard to say. So on top of this just being a species that's worth conserving, it's also a species that we have responsibility to make sure we're maintaining, because the declines that we're seeing are due to human interference. And so this and kind of every other species, I feel, definitely has value and should be conserved, and that research and monitoring should be done to do this. And one quote that I came across that I really had appreciated is just very bluntly, biodiversity must be documented before it can be conserved. And what the author here was discussing was how if we're not armed with the information to either care for the species, the background information about what it is, and the knowledge of its life up to now, we will not be equipped enough to take care of it from here on out. And that's everything. Thank you very much.
Awesome. Yeah. Thank you, Katie. At this point here, we are going to move on to the question period. I don't know if there's any questions in the chat here. Oh, yeah. So feel free to ask questions in person now, or add them to the chat and we'll ask them directly here. And yeah, Katie, you did very well. Great presentation, especially your last statement there about the value of the species. I feel like we should have that written on our door outside. No, it's fantastic. And you know, you hear that all the time. No question. All right. I have written down many questions, so I'll just go ahead and ask one of mine first. So what would you say are the biggest threats to prototype Quillwort in Nova Scotia and Atlantic Canada at the moment nowadays?
That is one of the big reasons why this kind of research is important, is that we don't 100% know. When we're looking at the map of where it's located, most of those lakes are entirely populated by cottages. And so right off the bat, I was like, oh no, that's it. Like, you know, people are cutting areas, there's pollution, there's all this stuff. And actually that has not been the case when I went and did the surveys though. A lot of the places that were either best maintained since 2004 or even have had slight increases are these very heavily cottaged lakes where people are taking care of their property. They're being good stewards. They're making sure that they're not polluting. They're, you know, there's not a lot of highway runoff because they're maintaining that kind of wood, the forested area around the lake. And so it's really hard to say. I'm sure pollution, acid rain, a lot of things like that are definitely big threats. But because we haven't had extensive monitoring or surveys, it's really hard to pinpoint any one thing. Yeah. So yeah.
Well, yeah. Question from the chat. Otherwise, I have another one I would really like to ask.
No, people are just saying thank you and great job.
Oh, thank you.
And so with it only being in 20 lakes, are the habitat requirements for the species, are they super specific? Do they need like a really certain type of habitat that, you know, this, or are there other lakes around that are just like it and there's just no prototype Quillwort in there and we don't know why?
It's a bit of both. It is quite a specific habitat. Like I said, like the cold clear water, it had the oligotrophic, so not a lot of nutrients. So obviously not a lot of pollution or runoff either. But this species really suffers with any competition. So if any other species start moving in, prototype Quillwort just dies right off. And so we did actually see a little bit of that where one of the lakes that had a business close by, and we were seeing some eutrophication and some pollution runoff, and then the lake wasn't clear and there was a lot more kind of lily pads and other species growing there. The prototype Quillwort was gone. So but one thing that's funny, a lot of quillworts were first kind of distributed by glaciers. And so you'll find 3 lakes in a row that are all identical habitat-wise, and it'll be in the first one and it'll be in the third one, but not the center one. And we're not sure why. So it's probably just because the spores weren't dropped there.
Right.
And so it's very particular and sensitive, but also just rare and random.
Good. So Carol has a question.
All right. Yeah, Carol, go ahead.
Hi, thanks for the presentation. I got observation first. You might like to look in Bell Lake, which is a glacial lake in Cole Harbor in Dartmouth, and it's very much the kind of lake you're describing. And I haven't seen in recent years how much development there is alongside it, but I think it might still be relatively looked after. So that's one thing, Bell Lake. Second thing, is it the only quillwort, or that we have here, or am I not hearing that right?
No, so we have about 5 species of quillwort. There's a few more that are listed that we never encountered. I believe there's also a river quillwort, but that's kind of more in Maine area, but it's listed as maybe being in Nova Scotia. But yeah, so there's a few different species, and they're all very, very similar. So
Okay. And the other thing was, any guess on how it's going to manage with climate change and global warming? It seems a very adaptable thing.
Ideally, it should do well, but like I said to Luca's question, we just haven't been surveying and monitoring it long enough to really know how it deals with these trends. One of the problems, especially with like a lake species like this, as kind of temperatures rise and everything kind of is slowly moving north, it can't move, it can't migrate, it can't get into another lake. So I feel like it probably has a very definite kind of threshold that once kind of temperatures have risen beyond that, it's going to be in trouble. So yeah, but that is another area though that we really would like to have more information about.
Thank you. That's really great to talk about.
Great question.
Mm-hmm. Yeah. Do you have any other questions here from our audience? I have one more. The Ex-situ plant in Acadia.
Yes.
I'm really interested in that. So how does prototype quillwort usually reproduce? Is it vegetatively or? Yeah,
that's another question, is that we don't definitively know. It can reproduce vegetatively, but it does produce spores, and that should be how it's reproducing. Right. So I was able to collect a plant, collect some sediment, collect some lake water, put it in a bin, and simulate kind of overwintering just by fluctuating temperatures and watching the light, but it stayed alive. So with that, we could hopefully collect spores and see if we can propagate those. We could try maybe introducing it into tissue culture. So that would be really, really exciting new kind of news, or maybe just seeing it as it grows, if we can divide it. So yeah, it'll be really interesting to see what we do with that. I have no idea where it's going to end up. So yeah. Yeah.
What are the populations like in like one lake with them? Like, does it vary a lot? Are they kind of consistent?
Yeah, a healthy, right now, the lakes that were looking the best had about 100 specimens that we kind of observe, because it's usually kind of a small area and little clumps around. There was one lake that had more quillwort than we could have counted, so that was a really happy surprise. And then there was one lake that was supposed to have a very, very large population, and we only really found one individual. Now that lake also was like multiple kilometers long, and so if it had drifted at all, we just weren't able to survey the whole lake. But yeah, so some lakes would maybe only have 20 to 50 individuals. 100 was a lot of the ones that seemed very stable and very consistent to the 2004 kind of records was about 100 specimens in the lake. So yeah.
Now you said they like clear water, low nutrients. That's not a common lake type, especially southwest Nova Scotia. So do we really expect it in very many lakes?
No, not at all. Yeah, and that's exactly it. And that's why, like information about hearing that this is like for Bell Lake, or that's how the new site 10 had been identified, is because those conditions of like, oh, hey, I know a lake like that, and let's go give it a check. So, so yeah, but also when the original surveys had been conducted, these lakes were like really extensively searched.
Okay.
And so basically every lake that we could think of that did have those conditions had been searched. But even saying that, like our surveys this summer, we had the site name, we had in some of them, it was just it's at this lake, but some of them would be like, it's in this alcove close to this part, and it still would take hours to find it. So the fact that it could have gotten overlooked is a possibility. So hard to say, but
where the eDNA would be incredibly useful.
yeah, if we can get that one to work someday, that would be a really great tool for the species. Yeah.
Who was your funder for the project?
I did an NSERC grant, but then also like the USRA, and then also just Acadia University.
Okay.
Yeah.
The province should be putting some money towards this.
Yeah, we all agree on that one. All right, fantastic. Any more questions popping up in the chat here? No, I don't think so.
No.
Yeah. Okay. Chat, you good?
Yeah.
All right. Well, with this, yeah, I would like to thank you again, Katie. It was really, really great to hear about prototypus today. I'm glad that I know nothing. I learned so much. Yeah, I learned so much. So that's really great. Thank you so much. And yeah, thank you everyone else that joined us today as well. At this point here, I would also like to thank our funders, the region of Queens that supports us and the seminar tonight. And as always, you can stay up to date with our seminars by following our Facebook account, our Instagram account as well. But yeah, you'll be able to watch our seminars, rewatch them on Facebook as well, or on YouTube channel, both on our YouTube channel and on Facebook. So you can watch them wherever you like. And yeah, we'll be hosting these seminars weekly throughout the summer, throughout July and August. So if you ever have nothing to do on a Thursday evening between 7 and 8, this is a really good spot to come and learn more about research that's going on in our area, conservation is going on in our area. And you can either be virtual or you can come and see us here at our lovely field station at 9 Merit Road in Kent, just right by Acadia National Park. And also you can get to enjoy some free tasty snacks. So yeah, with this, I would like to wrap it up. Again, thanks everyone for coming tonight. Hope you stay well, and hope to see you all soon. Bye-bye.